Hello, everyone. Thank you so much for being here today. It looks like we have people tuning in from all over the world. I see people from Abu Dhabi, from California, from Oregon, BC, Italy. Definitely tune in at the chat box that's at the bottom of your screen and let us know where you're tuning in from. Seems like we've got people from Washington coming in as well. Thank you so much for being here. And thank you to the internet, which is this mycelial web connecting us all. It's amazing that we are able to gather here today. From Scotland as well. So beautiful. So we are very excited to present this new session in the series, A Tribute to Terence, a celebration of life and legacy. 2020 marks 20 years since Terence's passing. And this year we honor his incredible impact by continuing the conversation of legacy for future generations. My name is Genevieve Wood, and I am a team member of the McKenna Academy of Natural Philosophy, the nonprofit organization hosting this event today. The McKenna Academy explores modern and traditional practices, ideas, and technologies that foster the understanding of nature and the cosmos. So I just want to give everybody a quick intro to the room, just so we know where everything is. As I mentioned before, there is a chat box that should just be at the bottom of your screen on mobile or on the bottom right. If you are on desktop, feel free to chime in throughout the event and connect with the community. Also, we would love if you could share this on social media with your friends. Again, on mobile, you'll see that there's a little share button at the top of your screen. On desktop, it is on the top right. And then on desktop, you should also have an option on the bottom left of your screen to share a short video clip. So feel free to do that. At the bottom of your screen, you'll see a green button that will link you to our website. Feel free to go check that out. You can sign up for our newsletter, check in on our upcoming programs, as well as make donations. This event is made possible by our generous supporters. Thank you so much. Right next to that green button, you'll see the option to participate in a poll. And we have a few different ideas on new events that we can hold. We'd love to hear you chime in on the types of programs you'd like to see from the McKenna Academy. Our email is also listed there. If you have personal suggestions, please let us know how we can best support. As a one last note, so we're going to have a live Q&A today after the program. And so you'll see at the bottom of the screen as well, there's an option to ask a question. So that's where you'll be wanting to ask your questions. You can also upvote questions from other people in the community. So definitely throughout, if you have any questions you'd like answered, make sure to upvote as well. As a note, we will be filtering through and answering questions based on relevance and community votes. So we'll just quickly recap the Tribute to Terence series so far leading up to today. We began this tribute series on April 3rd, honoring the 20th anniversary of Terence's passing, with an incredible run of fireside chats between Dennis McKenna, Terence's brother, and Terence's close friends. In these fireside chats, we welcomed on Bruce Damer, Luis Eduardo Luna, Paul Stamets, Wade Davis, Rupert Sheldrake, and Ralph Abraham. Following up on this amazing group of thought leaders, we've begun to welcome in a wave of younger change makers, really strengthening the bridge between generations as we weave a sustainable legacy for the future. After a fireside chat with Dennis McKenna, Seth Warner of Myco Rising and the San Francisco Psychedelic Society offered a free mushroom cultivation class. So you can access all of the replays from these previous sessions of the tribute in the same event link. There is, you'll see that there's the name of the session at the top and then the word more, ... If you click on that, it should bring down a drop-down menu and you can access any of these talks through there. Definitely do so. There's some inspiring and educational gems in there. So we are so excited today to welcome on Dennis McKenna and Merlin Sheldrake in a lively program celebrating Merlin's newly published book, Entangled Life, How Fungi Make Our Worlds, Change Our Minds, and Shape Our Futures. We'll have a few different segments of the event followed by a live Q&A. So definitely make sure to ask those questions. And without further ado, I am going to welcome on Dennis. Dennis McKenna is a renowned ethnopharmacologist focused on the study of hallucinogenic plants. He is the principal founder of the McKenna Academy of Natural Philosophy, as well as a founding board member of Hefter Research Institute. He was a key organizer in the Ayahuasca Project, and he also organized the ESPD 50 conference, as well as many other groundbreaking projects. His work has been foundational in the scientific exploration of ayahuasca and many other realms, both in modern science and the progression of culture. Thank you so much for being here, Dennis.
Thank you, Genevieve. It's a pleasure to be here. Do we have an echo?
I'm not hearing an echo. Are you hearing an echo?
I don't hear it anymore. I don't know.
Wonderful.
Anyway, it's always something. It's always at the last minute, right?
Definitely. Yes. Well, I'll let you take it away and introduce Merlin on. Thank you so much.
Thanks, Genevieve. I appreciate that. I want to thank my support, my assistants here. They're like the mystical sisterhood, Genevieve and Christina. They do a fantastic job of making this go smoothly. And I've been cautioned not to move my frame, but I did want to draw attention to my commemorative T-shirt here, which I washed and cleaned just for the occasion. So, I hope you can appreciate that. And I'm very happy to introduce Merlin Sheldrake. He is an amazing young biologist. He is the son of Rupert Sheldrake, who is an old friend of Terence's. They did many events together and wrote books together and so on. And through that association, we had Rupert on a few weeks ago in the part of this tribute. But Merlin is very much his own person, and he's a brilliant biologist. And with that innate sort of childlike curiosity that, in my opinion, I think characterizes great scientists. And he is not only a good scientist, but he's a good communicator. So I'm really pleased to have him here. I guess I should read his longer bio just to give some background. Bios often don't do justice, but he is a biologist and a writer. He has a background in plant sciences, microbiology, ecology, and the history and philosophy of science. That's a good thing. Study the philosophy of science if you're going to be a philosopher. I've often urged people to do that. He received his Ph.D. in tropical ecology from Cambridge University for his work on underground fungal networks in tropical forests in Panama, where he was a predoctoral research fellow of the Smithsonian Tropical Research Institute. You're a keen brewer and fermenter, and you have been fascinated by the relationships that arise between humans and more than human organisms. Certainly fungi fall into that category. His book, Entangled Life, How Fungi Make Our Worlds, Change Our Minds, and Shape Our Futures, was published in North America in May and will be published in the UK and other European countries this fall. So Merlin, welcome. And it's a pleasure to see you once again.
Thanks for having me, Dennis. It's great to be here.
So I guess since we last spoke, the book has been released and you're quickly becoming a superstar in this area. So what's happened since we last talked? You must be very busy handling these kinds of events.
Yeah, well, it's quite nice actually with the lockdown. It means I don't have to travel around from place to place. Every now and then the long arm of America reaches into my living room. And,
yes, ours too, unfortunately. Yes.
And then it reaches out again. So, and then things return to normal. So nice time for reading, but a lot of talking for sure.
Yes, yes. There's a lot to be said for the lockdown. I mean, I don't want it to go on forever, but we've learned a lot about the power of virtual media and how to reach out to people using these tools. And so, that part's been very gratifying. And I'm glad it has been possible to continue with our academy programs in the virtual space. So we don't want to go too long, but I just wanted to discuss with you one of the topics I wanted to raise is that this is part of the tribute to Terence McKenna's legacy. And as you know, he passed away 20 years ago in April of 2000. It's hard to believe it was that long ago, but indeed it was. And I wondered if you personally knew him back in the day when he was doing events with Rupert and Ralph Abraham and other folks. You must have been very young at that point.
Yes, well, Terence died when I was 13. So
okay.
We had quite a lot of time before that, though. I always associate him with these conversations and happenings that were always a little farther than I was allowed to go. There were little clusters of trialogues which happened everywhere under trees, and I always wondered what was being discussed.
Right.
One time when we were on the Big Island in Hawaii, staying with Terence, we were having a picnic in a lava field, and Terence and my father and Ralph went off into this lava cave just to check it out, leaving me and my brother and my mother, Jill Puss, and we waited and waited and waited, and the level of worry rose and rose and rose. And eventually they came out over an hour later, and it turned out that they'd gone into the cave and fallen into an impromptu trialogue while there.
I see.
This is often how I thought of Terence and what Terence did when I was young, this being out of sight and out of reach, just further than I could go. So I was always quite intrigued by what he had to say. Of course, his stories were very amazing. He used to tell us in his sing-song voice, which I imprinted on.
Yes, he was a very excellent storyteller. The Irish gene, I think. He was a raconteur, and he could make anything sound plausible. No matter how crazy, he just had a way of delivering it. And I wondered if you ever got a chance to read his book, Food of the Gods, and if that was any influence on you.
Well, yes, I've read parts of it, and I've heard him talk about the ideas he presents in that book, for sure. The stoned ape hypothesis being one of them.
Yes, I've been rereading the book because there's a new edition coming out, I think initially in the UK. Another company has taken it up, so they've asked me to write a foreword for it, and I thought I better go back and take a look at it, and rereading it, two things really stand out for me. Number one, how incredibly timely it is. What he is talking about, what he was talking about in the early 90s, and the themes that he highlighted about climate change, the crisis faced by our planet on so many levels, is just more relevant than ever. I guess the difference is that 30 years ago, we actually may have been able to do something about it. Now, 30 years later, our options are much more limited. I wouldn't say there is no hope, but I think the window to take serious action is closing. And the other aspect of it, he spends about the first half of the book talking about what I think has unfairly been called the stoned ape hypothesis, and the idea that the early association of hominids with psychedelic mushrooms, particularly psilocybe cubensis, may have influenced the emergence of consciousness and self-reflection in our species at a very early stage. And because it's called the stoned ape hypothesis, it's very easy to dismiss it. And many people have, well, that's just ridiculous, you know. And actually, it's not ridiculous. And the more, as I go over it again, his argument is very reasoned and very thoughtful, and in my opinion, quite plausible. Do you have any reflections on that?
I agree that it's plausible that psychedelics will have left a mark on the evolution of the human species, whether culturally. Culturally, it seems obvious that they would have done. Just look at the 20th century. We can see a huge imprint of psychedelics on the evolution of human culture over that short period of time. So it's not a big reach for me to imagine that stretching back much further. I do wonder whether, when Terence described this with relation to the emergence of our enormous neocortexes, and when it comes to the brain size piece of the argument, I do wonder, what about Neanderthals? Because Neanderthals had larger brains than us, so presumably there would have been a stoned ancestor of Neanderthals as well. And then, how many times did this happen? Of course, if it's as plausible as Terence makes it sound, then you'd expect it to have happened many times. And I wonder why there are maybe not other primates that have upgraded as drastically as us through the consumption of psychedelics. So there are these intriguing questions. So it strikes me that perhaps there is this period of human evolution some people describe as the cognitive revolution. Some people describe as the emergence of behavioral modernity around 70 to 50,000 years ago. And this was after we'd acquired our large brains. But our behavior started to change. Art emerged, or at least the evidence for art emerged, complex social organization, commerce, etc. And perhaps there could have been a psychedelic input to this radical cognitive transformation. That I find easier to believe than this giant swelling of brain size, the neocortex hypothesis.
Yeah, I couldn't agree more. I think the notion that exposure to psilocybin somehow, you know, we had, back when he wrote it, there was a term that was not very widely used that is now very widely used and perhaps overused, this idea of epigenetics. You know, that environmental influences can feed back on the genome and influence gene expression. And that is plausible to me. I think you actually put your finger right on it. You can't say any given group of primates, if they're eating mushrooms, are destined to become intelligent. I don't think it works that way. I think it's a unique combination of their situation, and it's quite possible. You know, if we look at how far back does this go? You know, it's clear that the precursors, well, nothing's clear about the distant past, but it's plausible that the precursors of Homo sapiens, which would be Homo erectus and even earlier species, evolved in this savanna environment. And the savanna environment in which there were ungulates, the mushroom would probably have been a part of that ecology. And undoubtedly, if there were hunter-gatherer primates in that region, they could not have not noticed it, you know, and undoubtedly consumed it. What the impact of that was is going to depend a lot more on cultural factors, I think. Absolutely.
And I don't think we should imagine that there's an unbreachable divide between cultural and natural factors.
Exactly.
For example, the discovery and domestication of fire, which you can think of as a cultural phenomenon, drastically changed our biological possibilities by granting access to energy source of cooked food, which allowed us to spend less time foraging and more time doing everything else. And so this porosity between our cultural and natural categories, I think, is fairly evident. So I don't have a hard time believing that psychedelic experience over time could feed back on our biological, physiological condition.
Right. Yeah, interesting that you bring up the topic of fire, because I developed a whole lecture on speculations on the antiquity of consciousness, which highlighted the discovery of the controlled use of fire as obviously it had to be one of the milestones in this evolution of consciousness, of the ability of the human species to actually impact their environment and modify their environment in ways that they, that enhance their adaptation. And fire was one of those. And the archaeological record, again, it's always spotty, but it's pretty clear that by about 2 million years ago, whatever the hominids were at that time, they were using fire in a controlled manner. And that was an incredibly powerful tool for them for a number of reasons. Number one, that they could actually use it in hunting by setting fire, driving these herds of antelopes or other similar animals into ravines, and then they could all feast. So it was a very potent tool for that reason. But another aspect of fire, once you have controlled use of fire and you learn how to make teas, you've essentially invented pharmacology. The precursors of pharmacology are the interface of chemistry and plants, or fungi in this case. So having these tools, that early on meant that intelligent hominids could essentially undertake pharmacological experimentation. Of course, the thing about mushrooms is they don't require any of that, that perhaps more than any other psychedelic, no modification is required. They're ready to eat. And in fact, they look succulent and delicious, and in fact are delicious. All it requires is the curiosity. And that, as we know, is a trait of our species that's led to really us becoming human. We've, out of curiosity and a desire to understand how the world is, we have invented art and science and technology. So, and I think, it's, there's no real evidence. You can't cite definite evidence that 2 million years ago hominids were using mushrooms, but you can't cite evidence that they weren't either. And it's most likely that in these environments, they probably were encountering these. And so the species may have had a relationship to mushrooms over more than 1 million years.
We know lots of animals do have relationships to mushrooms, and some animals actually prepare mushrooms. There are certain types of squirrel that find and deliberately dry mushrooms to cache them for later consumption. So the idea that we would the mushroom use, mushroom eating, and mushroom preparation go way back beyond even primates.
Exactly. And if you happen to be a primate with an already complex brain, or a brain that is undergoing rapid complexification, then the mushrooms could have been an epigenetic stimulus to that. As we developed language and, the visualization centers, all of these things in the brain that sets the hominid brain, especially the Homo sapiens brain, apart from other hominids. Other hominids do not have these large areas of neural real estate, you might say, devoted to language and the visualization of abstractions. And that's always what I've claimed when it comes to the stoned ape hypothesis, that they're teaching tools. They were ways that hominids learned to use these complex brains that they had to create an interior space. The brains may have evolved, their complexity may have been in part in response to environmental influences, the ability to have good visual acuity and very fine-tuned manual dexterity and that sort of thing. But on top of that, and in addition to it, it gave us the ability to essentially link interior abstractions to meaning, to symbols. And that's the essence of language right there. And language, of course, as we know, is something that is unique to our species. So
I'm not sure. I take issue with the uniqueness of the language to humans. And the more we learn about other animal behavior, the more we realize that sophisticated language exists in all sorts of, and I mean, birds are an obvious example, and whales and dolphins are another example. And so I think language is actually much more evenly distributed across the animal world, and even one could argue they're beyond the animal world. So I wonder, this somewhat undermines the psychedelic hypothesis, because if mushrooms are required for human language, then if we find language in all these other species and they don't seem to have used mushrooms, then either we have to massively enlarge the stoned ape hypothesis to become the stoned bird hypothesis, the stoned whale hypothesis, and then we have to wonder where they found mushrooms, or we can admit that language arose without the help of psychedelics. And so, for me, this is a bit of a pressure point on the theory.
Right. On the other hand, yes, birds, probably whales, other animals, we can't really speak to that. We know that birds have languages, but language in such species kind of falls short of, Shakespearean sonnets, for example. I mean, language is something that in primates, in us, is overexpressed in a certain way. I mean, no other species, maybe I'm committing speciesism here, because we can't really understand, these animal language, but the presumption is that no other species has the complexity of language that we have. So, but then on other non-human levels, perhaps it does. I mean, for example, plants, plants communicate with their environment through chemistry. And I've often said, chemistry is the language of plants. Anyway, I see Genevieve is here. She's about to cut us off here. Anyway, we can take this up later, but yeah.
We'll pick it up again.
Infinite possibilities for speculation. So it's a space we like to be in. So
most definitely. Thank you both so much. I feel like that short intro contained so many different points to continue research. So I'm looking forward to listening again and kind of branching off into all of those areas. We are going to move into the next segment of the event, which will be an illustrated podcast. So Merlin and Dennis, I'm going to bring you off screen and I will bring on the podcast.
Okay.
So this is an incredible discussion between Dennis and Merlin discussing Merlin's new book, Entangled Life. And so I'm just going to switch over to the video if it takes a moment. Thank you so much for your patience.
Merlin, welcome to this podcast. We don't even have a official name for it, but I guess this is a McKenna Academy podcast. I've been thinking of starting one called The Curious Monkey. So if we do that, this will be the first one, but we will get it out there. So tell me a little bit about yourself. I know you as the son of Rupert Sheldrake, whom I know more or less well. Very much respect him, and I imagine it's interesting to be his son because he's a very interesting person. I'm sure he led you down many primrose paths in your day. So tell me and the rest of the listeners what led you into this and led you to write this book.
So the topic of the book is fungi and the lives of this neglected kingdom of life.
Right. I should put it up there. Yeah.
The expository subtitle, How Fungi Make Our Worlds, Change Our Minds and Shape Our Futures. And so it's a big cluster of topics and interests. And I suppose I got into it in a number of ways.
Mm-hmm.
I studied biology and plant sciences at university. And so if you're studying plant sciences, you can't go too far without running into fungi for reasons we'll come on to. But before that, I've always been interested in transformations. How do things change? How do things rot? How does a solid dog become soil? There are piles of leaves in my garden growing up, and I'd bury myself there and try and feel them, this pile of leaves shrink, and feel it as it disappeared into the soil. And this question of how things change, how things transform, has always interested me. And again, one can't deal with that question for long without running into fungi, given that they're such prodigious decomposers and composers. So, but symbiosis, the concept of symbiosis, was a major gateway for me to the fungal world, because I became more and more interested by these intimate and ancient alliances formed between quite different organisms, ranging from in distant, very different branches of the tree of life. And this was what got me into the relationships between plants and their root fungi, their associates that live in their root, mycorrhizal fungi.
Mm-hmm.
And that's what took me onto the journey of my doctoral research, which was looking at these fungal networks underground in Panama and how plants and fungi relate with each other and how those relationships shape the wider ecology of forests.
Mm-hmm.
And so, so symbiosis was a big way in, and then it's a branching, winding rabbit hole. You can't think about fungi for too long without thinking about other types of fungi, and other types of fungi lead you onto other types of fungi, and eventually you find yourself enmeshed in this peculiar, curious world. And I still haven't emerged. I don't expect that I'll emerge anytime soon.
Mm-hmm. Well, that's very interesting. What this book really sort of brought to the foreground for me was those things, this idea of symbiosis, and also fungi. The impression from your book, and also my own impressions, are that fungi are like the great neglected fifth kingdom of life. And yet they're at the basis of all of it. I mean, my training, my background is in higher plants, in plant chemistry, and that sort of thing, and to a degree in mycology. But your book makes clear the impressions I come out. In my public talks, I often am fond of saying, the monkeys aren't running things. We're not running things. And sometimes I proclaim plants are running things because of their ability for photosynthesis, to bring energy in from the cosmos and use that miraculous process to create complex organic life. But then your book makes clear that actually the fungi are running things. the one that nobody notices, I mean, they're everywhere. They're everywhere in the soil. They're inside of us. They're inside of plants. And it seems clear that without fungi, plants would never have, they certainly wouldn't have migrated out of the sea. I mean, the fungi were on land first, and that facilitated that process. So I think maybe the fungi are running things. If we, I mean, it's all a symbiotic relationship. Everything is running everything. But if you had to choose a kingdom, that was really fundamental, I think it would have to be the fungi.
Yes, well, I mean, their neglect is part of my motivation for writing this book, of course. And the focus on fungi and their roles and behaviors in this book is not to suggest that other organisms, other types of organisms aren't important, but it's more to level up, level the books a little bit so that we can approach these questions in a more symmetrical way. Of course, it does depend on your perspective. As a professor I know who says that plants are what happened when fungi grow. When trees are what happened when fungi got smart enough to grow them. The idea that these organisms underground got smart enough to grow plants. And in some sense, you can think of mycorrhizal fungi, which are these root fungi that extend, lace outwards from plant roots into the soil. You can think about these fungi as cultivating their plant partners. Of course, you can think about their plant partners as cultivating the fungus as well. And this is where we enter this lovely two-directional process and two-directional identity.
Right.
These organisms co-constitute each other. They also have this two-directional flow of nutrients and resources. So I enjoy playing with those shifts in perspective, and I think it's very healthy for us to do so. And it helps to lead us outside of the ruts of our perspectives and thinking about humans and ourselves often as the center of things.
Right, right. Well, yes, because we are not, and it's not only that plants may cultivate fungi and vice versa. It seems that plants and fungi may cultivate humans in a certain way. We do tend to think of ourselves as the pinnacle of evolution and, at the center of evolution of life on Earth. But it's a very anthropomorphic view. And the more we learn about life, the more we learn that, we're not necessarily the most advanced species. And certainly, I think in light of what's happened over the last few decades, the last few centuries, we have to question whether we're even the smartest of species. And I guess it depends on how you define smartness. But, humans are notable for making extremely stupid decisions. So if intelligence in a species is exemplified by their ability to optimize their conditions to thrive, to interact with their environment in such a way that they thrive, I think plants and fungi have humans beat hands down because this is what they do. And the other thing that comes out for me about this was, as you say, symbiosis. Under the older model of evolution, we think of competition and survival of the fittest, and if one species thrives, another one must lose. That's not the way evolution works most of the time. I think it's much more about symbiosis, collaboration, these interactions between different phyla, fungi and plants and bacteria and animals. That collaborative symbiotic relationship is what drives evolution. And provides a model for social models as well. So,
yeah, I mean, it's interesting to think about how a novelty arises, because of course the survival of the fittest still is true, because by definition, the fitter organisms are those that survive. So right. It's not that's an outdated notion, but I think that we've just entered a larger room, and so. What makes an organism fit is not just ruthless, unmitigated competition, conflict, but that fitness can be generated by symbiotic interaction and collaboration, complete with competition and cooperation and conflict. Collaboration is I think of it as an alloy right. of all those things. And like a family, and we use this idea, you can have a family fraught with tension, but it still functions as a family, or a jazz group. You can have Charles Mingus, totally sick and tired of Charlie Parker coming on the stage, jumped up, can hardly hold his horn. They play some amazing music, but they are not getting along in any kind of way that we'd recognize. This is not a harmonious relationship, and yet they can generate an astonishing performance. So collaboration, I think it was this alloy, and the larger room that we're in, so these different sources of novelty. So yeah, so it's nice to have competition and conflict watered down, have it diluted by these more collaborative elements. And I think it really changes the way that we understand both ourselves and the world we live in.
Yes, I agree. Yeah, competition and fitness index and all this is just a small part of the picture. That's the thing. It is one strategy that these organisms can use among many others. And the symbiotic strategy is clearly a winner for many species because mutual species benefit. Yeah. And it does raise the issue about intelligence. I mean, it seemed, and of course, this, you've written about it beautifully in this book, the idea that these mycelial networks and these like forest associations, forest size associations between plants and fungi, I think brains, the idea that you have to have a nervous system to be intelligent, that's been knocked off its pedestal in the last few years as we've looked into plant behavior and plant intelligence. And it's not that you have to have a nervous system. You have to have highly networked systems, what used to be called neural networks, except they're not neural. They're, I would say, hyper-connected networks. And that's what fungi are. That is basically what they are, is these incredibly connected networks. That is like the whole of the biology of them.
Absolutely. I mean, I like to think of ourselves as, do we get around and we are able to do what we do by containing these flexible networks of neurons that ceaselessly remodel themselves. And fungi are flexible networks that ceaselessly remodel themselves. So they just do away with the rest of it. Pure flexible network. And of course, slime molds, which aren't actually molds, they're not true fungi, but they've done a lot to help us move out of this narrow paradigm. And they're so astonishing because they can solve quite complex spatial problems. They're just one giant cell. They don't even have lots and lots of different cells and that kind of organization. So they really blow the paradigm out of the water.
Right.
I have a friend who keeps a stable of slime molds, and he likes to talk about their intelligence by comparison with his own. And he makes the joke that he gets lost in his local IKEA stores and he can't ever find his way out without asking for help from an assistant. So he got the floor plan of the IKEA store and built it into a maze and put his slime molds in it, released them in the IKEA store maze, and watched them rapidly find the quickest way to the exit, much faster than him. So he says in this regard, they're definitely smarter than him. And it's a helpful goof, I think, to think about the way that we manage our spatial problem solving and to directly compare it with these organisms that do it in a very different way.
It suggests other ways of thinking. Yeah. Well, this is and this theme, sort of is threaded throughout the. So are you doing research now, or are you still in the academics, or what are you doing after you've written this book, or are you just waiting for the masses to notice it and then you'll be a superstar?
I have no idea what will happen with the book, but I'm not quite sure what I'm going to do afterwards. I have some research questions that I would like to pursue. I'd like to look into this question of fungal signaling using these pulses of electricity, these action potential like impulses, which has been astonishingly under-researched. I talk about this in the book quite a lot, but Stefan Olsen, who discovered in the late 90s that if you stick microelectrodes into some fungal networks, of microelectrodes of the sort that you would use to investigate animal brains or the nerves in animal nervous systems, you can find rhythmic, regular spikes of electrical activity passing down these long fungal cells, these hyphae, they're called, and they're excitable. If you put a block of wood on a wood-loving fungus, then it will respond by increasing the frequency of its spiking. And this is fascinating, of course, because it suggests that, you have tip-growing, elongated, electrically excitable cells signaling using complex, potentially very complex networks of electrical activity. But it wasn't dealt with, it wasn't picked up, no one ran with this idea, and there's some people working on it now, but it's really crying out for further investigation. So that's something I'd like to pursue for sure. How exactly that will happen, I'm not yet sure. But
yeah, well, I think that's the cutting edge right there. Or perhaps we should say the hyphal tip right there. Because from the way you describe it, fungi are they're like externalized neural networks. So call them provisionally externalized brains in a certain way. There are these hyper-connected networks that use both electrical conductivity and also, of course, chemical signal transduction mechanisms like neurotransmitters in brains or plant hormones in plants. They have it both. They have the chemical and the electrical. And the electrical communication in fungi has been not well investigated. It's a whole new concept. But I think the fact that both these things are going on, they're like externalized nervous systems, not confined in a cranium. And that's just incredible. And the way that they communicate, this is rather amazing. One of the things that, as I got through the book, I started to look at some of the notes, which are just as fascinating. You've written a very multi-layered book here, but something that came up for me in the notes I was thinking was about how differentiation into specific structures, usually like in mammals, involves many different systems, and there's bone, there's cartilage, there's nervous system, there's skin cells, there's all these things. We see fungi able to create these beautiful structures, the fruiting bodies, and yet it's all made from hyphae. If you cut into a mushroom, you don't find differentiated organs, you just find more hyphae. And this is rather remarkable. And I have to wonder about, how that, where is that template? Where is that? Obviously in the genetics somehow, but, I have to wonder about morphogenetic fields, the idea that your father proposed years ago, that I think has great merit and is, again, marginalized in science. But usually what's marginalized is something that should be looked at further. What are your thoughts? How do the mushrooms or the fungi, how are they able to go from a mass of tangled hyphae to these highly structured beautiful things, the fruiting bodies? How does that happen? You have any thoughts on that?
Well, this really is one of the biggest questions, the biggest open questions in mycology. Morphogenesis in outside the fungal realm remains a big mystery. There's big advances being made now with bioelectric fields and how these bioelectric fields pattern the development of organisms. But morphogenesis is a big, hard problem to crack when you're dealing with animals and plants. In animals, you have much more ordered forms. You have
clearer protocols of development. You know, if you're born with 2 arms, you'll end up with 2 arms short of an intervention. But when you have these decentralized organisms with very fluid bodies without body plans, then the question of how they generate organized form is a real puzzle. So it's interesting you mentioned morphogenetic fields. In fact, this concept was first invented with regard to mushroom form by Alexander Gurvich, who is a Russian developmental biologist, and he studied mushrooms, and he puzzled over them because he said, as you say, you know, you cut open a mushroom, you see that it's made of hyphae, tousled like uncombed hair, he said. And how is it the case when you have an animal body, you have order and form which is patterned right from the beginning? You know, when symmetry is first broken as an embryo, before the development of an embryo, from there you have these various axes of development, which then turn into these big organizing axes that we see in our bodies when we're mature. But with mushroom, you have order arising from less organized parts. You don't have order arising from ordered parts, which is what you have in an animal developmental story. So how is this the case? And so this is why he proposed morphogenetic fields, because he felt that there must be some organizing principle which wasn't encoded in just a previous organized stage. And so mushrooms were the gateway organisms to the idea of morphogenetic fields. This idea, of course, is being picked up and used in all sorts of areas of biology today, and lots of people have different ways of thinking about it. Michael Levin, who's doing very amazing work on the subject at Tufts University, he talks about morphogenetic fields as the rich fields of information in which all cells are bathed, constantly bathed in this rich field of information, whether chemical, electrical, or otherwise. And so mushrooms pose this riddle at the highest level, and we're no closer to finding out how they do it. So it remains this big, the staggering mystery. I mean, we understand so little about fungal biology, and we don't really understand how they elongate. You know, the very basic mechanics of their elongation and their growth.
Right.
We have some intimations, but there's a lot of very basic questions that we're unaware of. So, yeah, I'm glad you enjoyed that Gurvich's riddle. How it happens, I mean, some people propose that if you have all these tips, growing tips, and they all change into different speeds and directions of growth at the same time, then you can have forms like mushroom-like forms arise. But how do they all change at the same time into the different developmental modes? One over here, one over here. You need some organizing field to make that happen. So, yeah, it's a big puzzle. Also because, unlike in plants or animals, developmental hormones, growth substances, haven't been identified in fungi. So we don't even know on a basic level how this might be coordinated chemically. But yeah, I mean, this is one of the fun parts of writing the book, is the notes. I thought of the book like having a party, and there are different rooms at the party in the book, different chapters, different spaces, some more active, some less active. And I thought of the notes as a kind of backstage bar, where you could go late at night, if you really wanted to talk for a long time with some other nerd in some shady corner of the backstage bar. So anyway, you find yourself in the backstage bar with Gurvich, which is fun.
Right. Well, yeah, there are so many different aspects to it, and it seems like just looking at you can't really consider this process of morphogenesis in fungi. I mean, inevitably, it reminds me of stem cells, and stem cells in animals are like these undifferentiated cells that could go in almost any direction depending on the stimuli that you might have. And fungal hyphal masses, in some ways, are just collections of stem cells in a certain way, or I think the term is pluripotent.
Even more than that, I mean, so stem cells are pluripotent because they can regenerate any tissue in the body, but a stem cell can't regenerate an entire organism.
Exactly.
So fungal cells can regenerate an entire organism. So we think of those as totipotent, which is even more powerful than pluripotent. And yeah, so but quite right. Yeah, these undifferentiated or these cells with vast developmental potential. From one fragment of a network, you can regenerate a whole network, which means that fungi are, in some sense, immortal. You could have an immortal fungal culture that just gets propagating again and again.
Yeah. There's just nothing else like them on Earth, and it's important that people be aware of them because, as you point out, as many people point out, they're under our feet. They're everywhere. They're inside of us, and they're just fundamentally important to life. I was fascinated. Really, I was fascinated by every chapter. I love the way that you take a thread and go with it, talking about lichens, for example, or yeasts. I mean, how many people even pause to reflect that what yeasts do? Many people don't even know that they are fungi. And yet the whole fermentation industry and alcoholic beverages and all this depend on these little critters, these single-cell fungal organisms, which have amazing capabilities. And now, of course, they're being used for genetic research precisely because they're so simple. No organism is simple, but they're being used for like metabolic engineering. I just saw an article which undoubtedly you've heard of, that some molecular biology research group in the EU managed to introduce all the biosynthetic genes for psilocybin into yeast. That's quite an achievement. I mean, I'm not sure what the practical application is, because psilocybes are quite able to make psilocybin. They don't really need to have it transferred into yeast, but I suppose if you want to grow it in fermentation vats and produce gigatons of psilocybin, which might be a good thing, then you can use these technologies. But you can grow many mushrooms as well. And that's something I also wanted to talk to you about was psilocybin, obviously. And mushrooms that could make psilocybin. Obviously, psilocybin is much more ancient than the human species is. There were basidiomycetes about 75 million years ago. Is that the approximate time range? Mm-hmm. Somehow or other, sometime in the course of fungal evolution, these things came up with psilocybin. They presumably did not evolve it for the delight of primates that were 10 million, 20 million years in the future. What do you think? Why did psilocybin show up in fungal chemistry? And what are your thoughts about what's going on there?
It's a really big riddle. Of course, people have puzzled over it for a while, and it ties into a bigger question of why plants produce the chemicals, particularly the drugs that they do. It ties into these ideas of secondary compounds or secondary metabolisms, where these chemicals which aren't crucial to the everyday maintenance of an organism, but they arise, they somehow, well, plants do this, they generate all this metabolic novelty. These chemicals and variations on a chemical theme accumulate in their bodies, and we don't quite know why. Some of them collide with animals, where they become a drug, they confound them, they kill them, they lure them. And so it's not at all clear why this happens. So in fungi, we have a similar question. Once a chemical becomes adaptive to a fungus, once it collides with an animal that it confounds or kills in a useful way to the fungus, then it will fall into a more conventional evolutionary story with natural selection. Right. But before that, how did it arise, and what purpose did it serve when it first arose? I mean, it's 75 million years ago. There's a long time before humans came along. So people are puzzled over this. There are two interesting developments in the history of psilocybin that have arisen in the last couple of years. One is the discovery that the psilocybin gene cluster has jumped between different fungal lineages.
Yes.
By horizontal gene transfer. So it's one of the few examples in the higher fungi of horizontal gene transfer, which is when you have genes that move between quite unrelated lineages without them having to interbreed with each other.
And non-sexual means of gene transfer. Yes,
exactly. So it's an everyday occurrence in bacteria, very adept practitioners of horizontal gene transfer. But fungi, they do it, but less so, especially in the higher fungi. It's hard to find examples. Psilocybin is not just one gene; it's a whole cluster of genes, and it makes it harder for them to jump between different lineages because there are more parts. It's a more it's a fiddlier piece of baggage, and so the fact that it jumped between different lineages and the fact that it remained intact when it jumped between different lineages suggested that the ability to produce psilocybin was useful to these fungi, because it wasn't clear that before this finding that it was useful. Maybe these fungi just produced psilocybin and it didn't do them any harm, didn't do them any good, but it just sat around playing a sort of Z-list role as a metabolic intermediate for thousands of years until it encountered other types of animals. But the suggestion is that it was useful to these fungi because it stayed intact as it moved between these lineages, didn't degenerate. And so, and actually it's evolved more than once independently, the ability to produce psilocybin.
I was going to ask you that. Yeah. So,
So what was it for then, 75 million years ago? And this was a time of fungi and insects have a long and complicated history. And you have insects like leafcutter ants or termites, which have ancient collaborative relationships with fungi. Leafcutter ants can't live without the fungi they cultivate. The fungi they cultivate can't live without the leafcutter ants. This is tens of millions of years old, same with termites. And you have fungi that possess insects, control their minds, and don't do them any good at all, and like Ophiocordyceps. Those are also tens of millions of years old. So fungi and insects, they go back. And the suggestion is that psilocybin originally arose in environments where wood-rotting, dung-rotting environments where there would have been lots of insects and lots of fungi in close contact. So the idea is that psilocybin evolved to influence insect behavior. And the question is, for what purpose? And initially, I mean, the conventional idea is that, yes, it's a deterrent. So insects would either their wits would be fuddled, or they'd lose their appetite, or some other bad, unpleasant effects that would put them off eating these fungi. But then there are problems with that because, after all, mushrooms are parts of fungi that fungi actually often want to be eaten, for their spores to be dispersed. So it's not clear that the deterrent hypothesis is very effective. Of course, there are also lots of insects that aren't repelled by psilocybin, and in fact thrive on psilocybin-producing mushrooms. So it's a hard theory for that reason too. So then there's the lure theory. So psilocybin was a lure for insects somehow. That a
lure, right.
That helps spread their spores. And actually, probably it was a mixture of both, because today you have mushrooms like the death cap mushroom, which produces the toxins which kill lots of animals that eat it. But for some insects that are able to develop resistance to these toxins, they have exclusive access to a whole food source. So you can have a death cap mushroom which is both poisonous and desirable to different species of animal. And maybe it was the way with psilocybin too. It's very hard to know. But the main thing that seems clear is that it was in some way adaptive for these fungi to produce it. That much we can be pretty sure of.
It must be, because psilocybin is not a cheap organic compound to produce in terms of its metabolic cost. Especially for a non-photosynthetic organism. I mean, I often say about plants, well, they're photosynthetic. They have energy to burn. They can, so that gives them the capability to create this huge array of chemical diversity and secondary products. Fungi don't have that, although maybe they do, because they can, form relationships with higher plants and get nutrients that way. But I would think metabolically, it's a more of a challenge for a fungus to make psilocybin. There's actually a cost there in terms of consumption of ATP and this sort of thing. And so it must have had, it must be useful. The fact that there's horizontal gene transfer, that, it's widespread, it shows up in places where you would never expect it to, like this relationship with the cicadas. Overall, Sheldon, I'm getting this picture. I mean, maybe, that fungi are like these hyper-intelligent organisms that are actually orchestrating the entire biosphere. They've formed relationships with plants. They've formed relationships with algae, which are plants, but probably predate the vascular plants. And then they have this whole aspect of controlling animal behavior. The insect, like Ophiocordyceps, and its relationship with ants. I mean, this is as creepy as it gets. This is science fiction, essentially, and yet it goes on, and that may extend. I mean, that may extend to even fungi's relationship with humans. With these monkeys who have accidentally or not developed these extremely complexified nervous systems, and psilocybin and psilocin just happen to be the neurotransmitters that hit that part of our nervous system that has so much to do with our concept of who we are and our destiny, our place in the world, and all that. So it's actually a plot on the part of fungi to dominate all of life, which is not a bad thing. I mean, I would say, go for it, right? I mean, but yeah. It's amazing to me that this behavioral modification thing is very interesting. You talk about how the Ophiocordyceps can infect the ant, and basically the ant, the fungus becomes the ant in a certain way, or the ant becomes this vehicle that the fungus can drive around and do what it wants to do. Well, maybe you could extend that to humans. I mean, it doesn't actually invade the nervous system, but it creates a hormone, you might call it, a chemical messenger that can establish this relationship with the mushroom, and it makes us want to consume it and grow it and all of those things, So, Terence used to say, he had a famous quote. He said, "We're involved in a symbiotic relationship with something that has disguised itself as an alien invasion so as not to alarm us." And it's a good quote, but the thing is, this isn't an alien invasion. This is distinctly a terrestrial invasion. I tried to make the case a few years ago at this seminar at Tyringham Hall, and your father was there. This idea that mushrooms were extraterrestrial. There's no way you can make that case. I mean, mushrooms are clearly part of terrestrial phylogeny, but that doesn't mean that they higher fungi developed psilocybin 75 million years ago, and it was useful for their interactions with insects and so forth. And then when we came along, it just repurposed it in a sense, or we discovered it. And when you form a symbiosis with a new species, interesting, novel things happen, and that's what's happened. We're actually living through this.
It's astonishing, isn't it? I mean, the moment of the history of the 20th century, and the role of psilocybin producing mushrooms in the history of the 20th century is astonishing. It goes from Richard Schultes publishing a paper on them in 1939, and then through Gordon Wasson, Timothy Leary, into the counterculture, into the whole psychedelic movement, that it just spins around. There were these gateway organisms that precipitated vast cultural change in the 20th century. Really puts the concept of gateway drug into perspective, and huge change in a very short time. And I think it's one of the really dramatic stories in human and fungal interactions. And it's just there for us to see it so close in time. We don't have to push our way through dusty records.
Something very clever of Mother Gaia. Maybe this is fungi's strategy for saving the world. In a certain sense, to change the behavior and the worldview of this most complex and most confused species on the planet, which would be us. So maybe that's what's going on. I also wanted to ask you: have you ever read a novel? There was a novel published by Brian Aldiss, who was a British science fiction author, and he wrote a novel which was published in the UK. It's called Hothouse, and in the States it was called The Long Afternoon of Earth. Have you ever heard of it?
Mm-hmm.
It's very interesting. It's one of the earliest. It was published in 1962 in the States, and it's a story of Earth in the far future when it stopped rotating, and it's gotten much hotter. There's very high levels of carbon dioxide, and there's a narrow habitable band in this stationary Earth, and the plants have gone completely nuts, and essentially that part of the Earth is a rainforest, and there's very few humans left. Whatever's left of human have become these small green, almost shrew-like entities, but they're still intelligent. And it's an amazing novel. But the relevance here is that in the novel, there is this fungus that actually drops out of the tree onto the back of one of the main characters, and the main character, and then puts Haustoria into his nervous system and forms like this frilly collar around him that he carries around. And in the novel, they call it morel. And I guess it looks like a morel, but it is, in the novel, it's it becomes a teacher of this person, and it's giving him information and essentially wisely counseling him in as the novel plays out. And it's just very interesting. It's one of the few books, science fiction or otherwise, I've ever read where one of the main characters is actually a fungus. and I highly recommend it. If you haven't read it, you'll love it. And
that's great. I think we could have more. I mean, I think a lot of the stories in the history of life, the main characters have been fungi, and if only we learned how to tell them. So I think that's a really sounds like a helpful text. And funny, it should be about these reduced mammals, because have you heard the series about the role of fungi in the evolution of mammals? After the last, after the asteroid that produced the fallout that destroyed the dinosaurs, and then you have the vast wipeout of tropical forests and forests everywhere because you have winter lasting for a long time.
Right.
Debris catapulted halfway to the moon, the terrible tsunamis, and all sorts, every disaster you can imagine. But one of the effects is to have a very, very, very long winter, and so of course plants die, and you end up with this global compost heap, rotting forests. And in this kind of situation, the decomposing fungi rise to the fore. This is their kind of, they become these top organisms because they've got so much to consume. And in this period of time, mammals had been around with the dinosaurs, but the dinosaurs were the big animals occupying those kinds of niches. And mammals were around, but there was wily, ratty creatures that stayed small and darkened between the feet of lumbering dinosaurs. The only way that they could exist, given all this the arrangement of niche space. And so then you have compost heaps, and you have all these fungi thriving. And people are always puzzled about how mammals rose into the fore and became the big creatures after that extinction event. And because mammals have this twofold cost, you have to spend all this energy keeping yourself warm. What a strange thing to do if you can avoid it. And also you have the enormous hassle of parental care, which is a huge cost. And so how was it that these doubly disadvantaged mammals, wily though they might be, could rise into this enormous niche space, which they then did? So one of the theories is that these fungi were fungi abound. All these fungal pathogens also abound, and organisms that don't elevate their body temperature under their own steam are very vulnerable to these fungal pathogens. And so mammals, by being as warm as they are resistant to many fungal pathogens, which is why bats, when they hibernate, their body temperature drops by a couple of degrees and they suddenly become very susceptible to fungal diseases. So we keep ourselves, so the theory goes, warm and thus resistant to this abundance of fungal pathogens. And by being resistant to them, could rise into this huge niche space in a way that the dinosaurs and birds couldn't, because they were susceptible.
Right.
So it's a fungal protagonist story, which is quite fun. But there are so many of them. As we talked about earlier, the migration of algae onto the land and the origin of land plants is another one. And
the whole story about lichens. I mean, your chapter about lichens is really mind blowing. I mean, I know Trevor Goward, having been a graduate student at UBC, and he's been around forever. So it was great to see him featured, and his perspective on lichens is just amazing. I mean, I have, as a biologist, I know that lichens are an association between a fungus and an algae, but the picture is much more fluid than that. I mean, it's like lichens. I think you made it. You said it correctly. These things are not simply symbiotic organisms. They're actually like mini ecosystems. And there may be fungal participants and bacterial, microbial partners and algal partners all in the same mix. And a lichen is not, it can take many forms, I guess, depending on what the constellation of symbiotic partners is. And yet do you have any thoughts on that?
Yeah, absolutely. Yeah, it's fascinating. And one of the big discoveries in lichens in the last few years is that there's a third stable partner in some of lichen lineages, a yeast. If you have the yeast there, then it gives these lichens the appearance of a whole different species as to if you have the yeast not there. So these participants in the relationship really make a difference. Toby Sprabilla, who did that research, one of Trevor Goward's former students, he's working now on bacteria, and he thinks that these bacteria aren't, because lichens are full of bacteria as well. Some people hypothesize that lichens are very important in seeding barren environments with new bacterial strains. They're these kind of they're small dosing organisms that allow these new barren environments to develop into young ecosystems. So, but he thinks these bacteria have developmental roles, that you need these bacteria to tie it all together. He says to tickle it in the right way, so that this relationship forms these complex structures rather than just a blob on a petri dish. And one of the funnest things with lichens, that the findings that I really like the most is very recently, 2014, researchers at Harvard did experiments with fungi and algae that didn't live together, that were free living, but by putting them in certain conditions whereby one could provide a metabolic service that the other couldn't, and the other could provide a metabolic service the other couldn't. They could reciprocally, together they could do something that independently they weren't able to do. So you had these new symbioses between fungi and algae form in 10 days. Less time than it takes a scab to heal. You have a soft green ball, the birth of a new symbiosis in a lab, in a flask. It happens that easily. These aren't the complex forms that we see in things that have been around for millions of years, but it's a start. And so there you really see that this is just waiting to happen. And of course, lichens have happened, evolved multiple times over the course of their history. And lots of fungi have delichenized. There used to be lichen forming fungi, but no longer. Some of them have relichenized over the course of, switched partners over the course of the evolutionary history.
Relichenized with different partners. Sometimes.
Absolutely, yeah.
Right.
And some are facultative lichen forming. They live as lichens or not, depending on the circumstances. It's just an optional lifestyle choice. But 1 in 5 of all fungal species form lichens. So this is not such an exceptional behavior. It really is. I think of lichens and fungi and algae as two marbles, the edge of a bowl. If you release them, they roll down to circle around each other. It's just waiting to happen. Seaweeds, most seaweeds are, again, relationships between algae and fungi, just like land plants. So land plants are a kind of evolutionary refrain.
So these are, the lichens are an incredible laboratory to understand this process of symbiosis, almost on a compressed time scale, because, as you say, they can form these symbiotic relationships, essentially create a new lichen in a few days of being in proximity.
I should say that these ones they made in the lab, they're not what we would call, they're not formal lichens like we would. They wouldn't designate, but they're a lichen-like principle, and you can imagine that maybe one day they would become a lichen. But but they're not formal lichens. But yes, it does. It provides this amazing glimpse, this window into the earliest days of this kind of relationship.
But given enough time and the right circumstances, those new symbiotic partners would develop into a lichen that would be stable, and because many lichens, I guess I guess you would expect that they would sort of be, I don't know what the term is, they would be facultative alliances that would then fall apart when conditions change. But many of them persist, and some are millions of years. Do you think that lichens, some sort of lichen-like organism, was the first land plants, and that then eventually that developed into plants with roots and so on? Is that how it took place?
It's an area of big speculation, and there are lots of different hypotheses. So some people have suggested that lichen-like structures were the first land-dwelling plants. The molecular evidence suggests that what we now call lichens didn't arise until after the earliest land plants. So it doesn't mean that you couldn't have a lichen-like organism that arose before land plants. And indeed, before land plants happened, you had cryptogamic crusts, formed by algae and fungi making a living on scorched, parched, UV beaten ground of dry land, which are hostile environments. So who knows what kind of fungal, algal alliances were formed then? And we might think of those as lichen-like, taking lichens to be a principle rather than a taxonomic lineage or a collection of taxonomic lineages. So it's possible. Some people say that plants are inside-out lichens, whereas lichens, you have the fungal form, the fungal organism providing the bulk of the body, and then the algal form, the photosynthetic partner, whether it be bacteria or algal, forming the beating metabolic heart. With plants, you have the algal form taking this external bit, and the fungal being inside, whether it's inside the leaves and the shoots or inside the roots.
Right.
So
it reverses that relationship where the bulk of the biomass is plant, and with the fungal partner, and lichens are are the opposite. And I've been fascinated by fungal endophytes, which I guess virtually every plant has. From the chemistry point of view, these are an unexplored realm of molecular diversity. You know, there's lots of interest in finding new compounds, potentially with medicinal properties and other types of properties. And it seems that we may have to rethink, you know, what we think of as this plant-generated spectrum of biodiversity or molecular diversity. Much of it may actually be the endophytes. And that's really exciting.
It's thrilling, isn't it? And of course, the ergot fungi that Hoffman used in the process, the derivation of LSD, they're an endophytic organism. And and Taxol, the multi-billion-dollar blockbuster anti-cancer drug, turns out to be produced not by the yew tree, but by the fungi that live in the yew needles. And but only in relation with the yew tree. You can't get Taxol to be produced by the fungus on its own, nor by the plant on its own. It's somehow a property of their relationship. But yeah, I think there'll be, as we discover more and think more about these endophytes, a lot of the drugs that we give credit to plants at the moment will turn out to be produced by their fungal partners.
It's a collaboration between fungi and plants that creates this molecular diversity.
And even more, I mean, there's a great study where it turns out that a tropical grass that lives in hot soils can't live in these hot soils without a fungus that grows inside it. But it turns out that there's a virus involved as well. So if you cure the fungus of this virus, then the plant won't grow in these hot soils. Neither will the fungus.
That's amazing.
The virus and the fungus in the plant, and only with this virus will the fungus support the plant's growth in this environment. There are more dramatic experiments with different grasses. There's one where there's a type of grass that grows in hot geothermal soils, and one that grows in high salty soils, high salinity. And if you cure them of their endophytes, neither grass can survive in their native habitat. But if you swap the endophytes, then the grass that growing in the hot soils will start being able to grow in the salty soils, and vice versa. And you
That's
can really
incredible. I mean, tell me this is not intelligence at work. You know, I mean, it's just rather amazing that goes on. And you're noting that yes, the ergoline alkaloids, the lysergic acid derivatives, are endophytic alkaloids. They come from a fungus. So, and you're probably aware that certain morning glories have these psychotropic properties. They have uses in traditional shamanic medicine and so on. You're probably aware of the work that has been shown that actually the morning glory ergolines are of an endophytic origin. If they're in the seeds, which endophytes often are, and but if you grow the seeds, if you grow the plant under conditions and you add antifungal compounds, they don't make ergolines anymore. You know about that.
Yeah, I mean, it seems to be it's a recurring pattern, you know, and so together, I think it's a common theme in symbiosis. Together, organisms can do things that when they're apart they just can't do. The relationship extends both of their reach. And with lichens, lichens are amazingly ingenious metabolically. Fungi are already metabolically ingenious organisms, but lichens particularly so. There are over 1,000 compounds that are only found in lichens. Unique lichen compounds, they're called. Sunscreens, antibiotics, dyes, various flavors. One of the ingredients of garam masala, spice mix, is a lichen. Humans have.
You mentioned that in your book. Yes. I love garam masala.
Well, you're a lichenivore, like so many others.
Indeed.
So, but one of the fascinating things is that in fungi that have de-lichenized, so that earlier in their evolutionary history they formed lichens, like penicillin molds. They've now de-lichenized, and now they don't form lichens. But there's lot of persuasive evidence that actually these compounds, potentially including penicillin, arose when they were lichens, and in this crucible of metabolic creativity and diversity that they had in this relationship together. And these compounds, such as potentially penicillin, persist as metabolic legacies of their lichenous past. And so we might see the imprint of this algal fungal metabolic creativity, even when we don't see algal fungal relationship living today.
And you could trace that maybe phylogenetically or through DNA analysis. Yeah, that's very interesting. Lucky for us, these fungi invented lucky for us they invented psilocybin, but lucky they invented penicillin and similar marvels as well. I mean, well, this is a fantastic conversation. I wish we could go on. I'm trying to keep it about an hour. We're already over. But tell me, where can people buy your book? I guess the usual places, or?
The usual places. If you want to support indie booksellers, which is great, then there's a place called bookshop.org, which is
bookshops.org. Okay.
Bookshop.org, and but otherwise all the normal places to buy books. There's an audio book, there's an ebook. But yeah, bookshop.org would be a good place to go. And it's available from the 12th of May in North America, and will be released in Europe in the fall.
Yeah, you've done a important bit of work here. This is really a significant book, and I think the world needs to know about it, and they need to know about you. You're doing fantastic work. Merlin, thank you for taking time to talk to me.
Well, thanks for having me. It's been a pleasure.
Okay, yeah, enjoyed it very much. We'll be in touch.
Absolutely. Thanks so much.
All right. Have a great day. Yeah.
Thank you.
Wow, that was a fascinating discussion. Thank you both so much. In this next segment of the event, Merlin is actually going to share an exclusive section of video clips. Very much looking forward to this. They're made from laser scans of roots collected in Panama and show the intimate symbiosis between plant and symbiotic mycorrhizal fungi. So thank you for your patience just while I bring Merlin on screen. I am so excited to continue learning about all of these amazing topics that were just brought up. Thank you so much again. All right, so we should be bringing Merlin on. Hi, Merlin. Thank you so much. That was incredible. I'm going to bring Dennis on now, and if you'd like to start sharing your screen with these incredible video clips.
Thank you. Let's try this. So,
okay.
Great. There we go. We've got the regress. So now, can you see this? Dennis, can you see a?
I can see it. Yes.
Great.
Can you make it bigger or not? Ah.
No.
Okay.
It's not.
Can't make it bigger with us
me.
on the screen, but we don't need to be. Yeah, this looks good. Everyone
Great.
can.
So.
Okay.
So I'll say a little bit about these videos first. What they are laser scans. They're made using an instrument called a confocal laser scanning microscope. And lots of microscopes, traditional microscopes, use light and they shine light through your subject. And you see using the light that's shining through the specimen. But laser scanning microscopes use laser rather than normal light. And what that means is you can focus on a very precise depth. You can focus on a precise focal depth. And what that means is that you can take slices through a sample using the laser. So you can scan the sample at a given focus, and you can scan again, focusing a little deeper. And you can scan again, focusing yet deeper. And so how you use these microscopes is to take a series of scans and then to overlay the scans and render a three-dimensional image out of these scans later on, after you've scanned the sample. So I was working for a while. I wanted to get inside plant roots and to look at this incredible symbiosis, the relationship between plants and mycorrhizal fungi. And these relationships are astonishingly intimate and inaccessible because they happen not just underground, but inside plant roots. So they're doubly shielded. And I got in touch with an amazing researcher and microscopist in Germany called Stefan Imhoff and his very gifted student called Magnus Rath. And together they'd been devising this new technique to use confocal laser scanning microscopy with a certain way of preparing samples that allowed a whole new resolution and depth with these mycorrhizal roots, mycofungus rhiza root. So I went to work with them, and Magnus is like a watchmaker, and with these skilled, precise movements and astonishing patience, and a very, very gifted human. And so with their help, I produced these scans of roots, which I collected in Panama, in the tropical forest in Panama. And so what you'll see here are these various scans of plant roots, and the plant tissue is rendered in blue-green, and the fungal tissue is rendered in red. These are artificial colors added later. But what you'll see is that you can turn down the plant channel to reveal the fungus within the root, which gives you this amazing sense of not just the fungus, but how the fungus and the plant tissue embrace each other, clasp each other, intertwine on the cellular level. So I'll commentate as we go through so you can see. So we'll start with this one here. So this is the root tip. And as we turn around, the plant tissue is now gone. And so you can see how the fungus is really a kind of organ, another organ of the plant. It is restricted to certain parts of the root. It's not just a free-for-all. It's very carefully and intricately managed within the root. And the tissues here's another one. You can see the fungus is also present on the outside of the root, because the fungus lives inside the root, but it ranges outside the root, and it laces through the soil where it forages for nutrients, which it then shuttles back into the root. Here we're actually going to take a trip inside the root. I'd always dreamed of doing this for a very long time, and I was so thrilled when it became possible to get inside the root. And there we go. This is something I always thought would be restricted to my imagination, but it's actually now possible. One thing I want to do with these scans, it's possible to use these data sets to produce 3D prints. And so I had this idea to produce these giant human life-size 3D printed models of these roots out of a kind of rubberized silicone, so that people could squeeze their way in and around these roots and feel what it was like to be in such close quarters with plant tissues. Here's another one where we're going to go inside the root as well. And here you can see this, between this central layer, these bladder-like structures. Those are where the fungal hyphae, the fungal cells, they grow inside the plant cells, and then they kind of pop. And when they pop, they release their contents into the plant. So that's where the exchange happens. Different plants have different types of relationship, and you'll see later a different kind of organ of exchange. But you can see there's this kind of transport plumbing type hyphae, and then there's these exchange type organs. Here we're going to go around the view from the inside out. And so, yeah, now we're going to now we'll go over to here. In this video, you can see here how these swollen coils are really they're situated right snugly inside these plant cells. And as the video plays, you'll see that more clearly. So this is intimate, not just fungal hyphae growing amid the cells, in and around the cells of the plant. It's not just inside the plant; it's inside the plant cells, and it's really hard to imagine something more intimate than growing inside the cells of your symbiotic partner.
So Merlin, is this all one species of fungus that you're looking at, or you're looking at varieties of root symbioses here?
So these are all one species of plant, and it's actually an unusual type of plant. It's what's called a mycoheterotroph, and these plants have lost the ability to photosynthesize, and they're pale white, and so these have quite a distinctive relationship with fungi, and their roots are more like fungal farms than normal plant roots, and they have quite specialized relationships with fungi. So in these images, you're seeing predominantly one sort of fungus. There are probably maybe 3 species or 3 different types you're seeing in these videos so far, but predominantly one. In this video, however, you see something different. What you see here is a different organ of exchange. This is called an arbuscule, or a little tree, from the Latin. And so this is a different type of plant. Here's the vascular bundle, the bundle of vessels that moves up the center of the plant stalk, through which the plant transports solutes in water. And the arbuscule is one of the very typical organs of exchange. And in crop plants, in grass ears, in many of the most prominent species of plants, you have arbuscules as the site of exchange between plant and fungus. So these organs really are, many of the nutrients that inside your body will have passed through an arbuscule at some point on their journey before they reached you. So here we're going to look more closely at an arbuscule, and I always love looking at the piping. We think of these fungal mycelia as webs or as threads, but really they're tubes. It's easy to forget about the space inside the hyphae, and so this is a good reminder that we're talking about these networks of tubes.
Mm-hmm. So this unique plant that does not have chlorophyll forms these symbioses. How selective is that? Can it potentially form symbioses with various species of fungi, or is there only one that?
It depends on the plant. Some of the plants are very specialized. This one is one of the very specialized ones. And these ones tend to form a relationship with only one type of fungus, although in the forest where I was studying them, they formed a relationship with the most prevalent fungus in the forest, which allowed them to connect to the maximum number of other plants. Some of these plants are less specific. On the whole, normal green plants tend to be fairly unspecific, and and so this is why we have the concept of the wood wide web, because plants are promiscuous. They can bond with more than one type of fungus, and fungi too are promiscuous. So this leads to these overlapping shared networks. So on the whole, these relationships, they're fairly open as symbiotic relationships go. And the images I've shown you are from this somewhat more selective example.
Are these symbiotes usually basidiomycetes, mushrooms, in other words, or?
Well, they're not mushroom forming. These ones are. Let me get out of here. They're infinite regress. There we go. So they don't form mushrooms. They're from the Glomeromycota. So it's a phylum of fungi, the Glomeromycota, and they're a very ancient phylum, and they produce spores without producing mushrooms, which is one of the reasons why they're hard for us to see. Some types of mycorrhizal fungi, the ectomycorrhizal fungi, produce mushrooms like porcini or chanterelle or truffles. But the Glomeromycota don't do that. So we have to examine them in different ways.
Do you think that the ability to form a symbiosis with the fungus, I don't know how to put it, but essentially enabled the plant to lose chlorophyll?
That's exactly right. So normally plants would give carbon, they would exchange carbon with their fungal partners, and they receive nitrogen and phosphorus in return, other mineral nutrients from the soil. So there's this two-way exchange, and with these plants, they seem to have worked out how to receive both carbon and mineral nutrients from the fungus. But because the fungus receives carbon from other plants, it means that this carbon that powers the life of the microheterotrophs is traveling from a green plant through the fungal network into the microheterotroph. And so it's exactly the fact that these fungi can connect to more than one plant that allows these microheterotrophs to survive. I think of them as hackers of the Wood Wide Web.
Exactly. Hackers of the World Wide Web. I like that. Yes. Very interesting. Very interesting. So essentially the fungus and the plant are involved in a co-conspiracy to steal nutrients from the rest of the forest. These hard-working photosynthetic plants that are producing.
Well, that's one way of reading it. I'd read it differently. But that's, no, one could see it that way. I don't think it's really very true. But the big question with these microheterotrophs is, are they giving anything back to the fungus? And so some people call them parasites. They call them, these hackers, freeloaders, whatever, or this pejorative language. Because it's very hard to see, when you're looking at the normal currency of exchange, which is carbon and nutrients, how they could be giving anything back, because they don't have a means to do so, because they're not photosynthesizing. So they must be cheaters. But it's possible that they're giving something back. They're returning the favor in a currency that is not nutritional. For example, by providing shelter in their roots, these cavernous root spaces, and the soils are bustling wilderness, and there's a wild west of the open soil. And if the fungus can have this shelter, this sanctuary in this root, then it's possible that that's a benefit worth paying for. So there are other things that we might not have taken into account. My father calls that the saloon theory of microheterotrophs, that these roots are a kind of saloon bar for the fungus, where it can take shelter and get refreshed.
Very interesting. Very interesting. All right. Well, thank you for. So this is work that came out of your work in Panama. This is what, and for the people that want to dig deeper and look at some of the published the scientific papers that have come out of this, where would we find those? Mycologia, or
there's a page on my website called Articles. Okay, and then that's a good place to start.
Okay, well, that sounds.
My website's merlinsheldrake.com.
Right, merlinsheldrake.com. All right, well, that's very interesting. Do you I think Genevieve is about to come back. We're a little bit past time, but not bad. There she is, right on cue. All right.
Wonderful. Thank you both. Those videos were gorgeous, and it's really amazing that you, when you were speaking about wanting to envision what these looked like as a younger person, and then being able to go through the process of creating them and sharing that with us is so beautiful. Thank you. So we are going to move into our Q&A section. And one topic we'd really love to hear about, Merlin, if you could maybe speak a bit more about fungal signaling. I know that you've mentioned this is a topic that you'd like to study more, and that it's really crying out for further investigation. So would love to hear your thoughts on that.
So there's a few ways into that question. One, on a basic level, fungi use chemicals to signal to the parts of themselves and to other organisms. So for example, if a fungus and a root, before they get together, before they can find each other amid the fuss and chatter of the soil, the plant root would emit signals that would attract the fungus, and the fungus would emit signals and also chemicals that are plant hormones, actually, that change the developmental process of the root. And so the root and the fungus attract each other and alter each other's physiology and developmental behaviors using chemicals. And so there's a whole way in which fungi use chemicals, as many organisms do, as coordinating, as a medium for coordination. But then there's these other ways that plants, for fungi can signal and communicate. And so one of the big questions in fungal biology is how that they stay in touch with themselves. These networks can sprawl over these large areas, and how can one part know what the other part's doing? And so there are a number of theories, and one of them is that the flow is important, because these networks are networks of flow, and this river of cellular fluid, the cytoplasm, moving through these networks from areas of higher pressure to lower pressure. This could be itself, flow could itself be a kind of signal, a developmental signal. Also, it's possible that if you change the pressure in one part of a network, you could feel it in other parts of the network almost immediately, because like a hydraulic braking system of a car, these hydraulically continuous networks are responsive to this kind of pressure. Change the pressure in one place, you'll feel it everywhere else. And then there's this question of electricity, which I think is really fascinating and totally underexplored. And so these fungal hyphae are electrically excitable and conduct these waves of action potential like impulses, analogous to those nerve impulses that pass through our own bodies and coordinate our own movements and thoughts even. So this is a very underexplored field, and the Stefan Olsen, who did this work, I love when he tells me the story. He says he went next door to these biologists